Welcome to today's episode where we delve into the molecular mechanisms that govern gene regulation and nuclear organization. Our guest, Dr. Anders Sejr Hansen from MIT, investigates how dynamic chromatin interactions and transcription factor kinetics shape gene expression in real time. By combining advanced live cell imaging, quantitative biophysics, and genomics, his research reveals how the genome functions as a dynamic self-organizing system. Join us as we discuss how this mechanistic understanding is reshaping our view of transcriptional regulation and its role in development and disease.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
And today I'm happy to talk to Anders Sejr Hansen from the MIT. Anders, welcome to the show.
Thanks so much for having me.
So, to ease into the topic and, to get to know you a little bit better, a question I like to start with is, how did you become interested in biology in the first place and then in pursuing a career in science?
Yeah, the short version is that I became interested in biology and pursuing a career in science quite late. I think it was only during my PhD. The slightly longer version is that when I grew up in Denmark, I went to school and high school in Denmark. I enjoyed math, chemistry, and physics a lot. But my primary school, it had some issues, and there were like a few subjects we were supposed to cover that we didn't cover, including biology and grammar and a few things. So when I came to high school, I realized that other students had taken a lot more. They'd studied a lot more in areas that I hadn't, and one of those was biology. And so I actually didn't really enjoy biology in high school. I thought it was very focused on memorizing things instead of understanding things. And that might just be how it was taught in my high school. But I enjoyed math, chemistry, and physics. And so I ended up studying chemistry at university. And I especially love organic chemistry because, understand a couple of principles and then you can just deduce everything by logic and reasoning. There's no memorization necessary. So I really enjoyed studying chemistry, but after my first year, I did a summer research project where I worked in total synthesis, and that left me a little bit disillusioned actually, because although I love studying organic chemistry, I found that, and I mean absolutely no offense to organic chemists, but I found that a lot of the research was focused on improving the yield of a reaction a little bit, or making a molecule to show that you could make the molecule, but it wasn't so focused on really fundamental questions. And so after that, I sort of thought, okay, maybe chemistry is fun to study, but it's not my core passion. And so I wanted to switch to biology. And because I was also very interested in math and quantitative approaches, I thought quantitative biology, systems biology would be the way to go. But my undergrad, we only had physical chemistry, organic chemistry, and inorganic chemistry. Those were the only three courses I had. So then I joined a chemistry program for my PhD that was very flexible. You could join any lab you wanted to. You could take any course you wanted to. But that also meant that when I arrived in my PhD, I had never taken a biology course. I didn't know that a plasmid was just circular DNA. And so it was a tough transition, but a very rewarding one.
So are you still...
And then, sorry, go ahead.
Are you still influenced by this education?
I suppose so. Yeah, I suppose so. It's hard to know what my view would have been like had I studied something else. But I I like to say that what we do now in the lab, it's basically all the sciences, more or less, except chemistry, in the sense that we do biology, physics, math, computer science. So the only thing I studied is kind of the only thing that we don't do in the lab now. But, one of the fun things about academic research and science, right, is that you can you can continue to learn throughout your career.
Yeah, that's very true. So for those unfamiliar, could you introduce yourself and your current role?
Yeah, so I'm an associate professor at MIT in the biological engineering department. I'm also a member of the Broad Institute and the Koch Institute for Cancer Research here at MIT. And my lab is generally interested in understanding the relationship between 3D genome structure and function. So how is 3D genome folding regulated? And how does 3D genome folding regulate functional processes such as gene expression regulation, especially by distal enhancers?
So when looking at your papers, I guess, when the interest in this started was when you were with Robert Tian and Javier Darzac, if I pronounce it remotely correctly. So there you focused on loop extrusion, loop structure, and loop stability. So how is loop stability regulated? And I guess the most important factors there are CTCF and cohesin.
Yeah, that's right. Yeah, so I've always been really fascinated by dynamics. My PhD was all focused on how you can use dynamics to encode information, a little bit like FM radio and so forth. And and I studied gene regulation in yeast during my PhD. And when I compare mammalian genomes and human, yeast genomes and mammalian genomes, the core molecular species are more or less the same. You have TF2D, Pol2, mediator, histone modifications, etc. But one of the most striking differences, at least to me, was that, the yeast genome is about 70% coding. The human genome is just a bit more than 1% coding. And yeast does very sophisticated gene regulation with a few hundred base pairs of a promoter. But in humans, you have these very distal enhancers. For example, the MYC enhancer in T cells is 1.7 million base pairs distal, right? So if yeast can do sophisticated gene regulation with 300-400 base pairs, why do humans need 2 megabases? So that was really what fascinated me at towards the end of my PhD. So I thought, let's try to understand why humans are so complicated at this level. And so, as you mentioned, I joined Robert Tian and Xavier Darzac's lab at UC Berkeley. And I was also very interested in single molecule microscopy. So this was sort of the two things that I really wanted to explore. There was looking at the dynamics of these proteins using single molecule methods. And the reasoning was, Hi-C is just an incredibly powerful technique, right? It was developed by Joop Dekker, Ernst Lieberman Aiden, and a few other people. And, it's revealed AB compartments, topologically associating domains, CTCF, cohesin loops, and so forth. And it's taught us so much, but it is a static method. And so one of the questions that fascinated me was, if we actually look at a single cell over time, how often is a TAT fully looped? How long do these loops last, and so forth? So that was what I thought was a missing piece that I wanted to study. And then what I focused on in my postdoc was the protein CTCF and cohesin. And so we used a variety of single molecule live cell imaging methods, super resolution methods, and FRAB methods to quantify the binding times. And so one of the things we found was that CTCF binds, has a residence time on the order of minutes on average in mouse embryonic stem cells, and cohesin has a residence time on the order of 20-25 minutes. And so if these proteins dynamically bind DNA and are also required for forming the loops, then it's reasonable to assume that the loops might not be infinitely stable. But that is, of course, a little bit indirect. Ideally, you want to look at the loops directly. And so that was one of the first things we wanted to tackle when I started my independent lab here at MIT.
Yeah, we will come to that in a minute, of course, because you also investigated the role of CTCF further in loop extrusion and how it can find its target regions, right? Because it might not be random.
That's right.
And it needs to get to the DNA. So how does this, I'm not sure if I remember this correctly, because we have talked about CTCF many times also with Joop Dekker and people like that. But there might not only be one CTCF at a site. So how does this clustering and the recruitment of CTCF to the right regions work?
Yeah, those are really fascinating questions. If I can take a slight detour, I actually think the question of the question of how transcription factors find their target sites is still one of the most fascinating things. And there's this classic experiment from 1970 by Riggs where they mixed DNA and the lag repressor. And then just measured the on rate of the lag repressor finding DNA. And what they found was that the K-on was extremely fast. It's 100 times faster than the Smoluchowski limit. The Smoluchowski limit is the fastest a reaction can be if, like, every single time two things bump into each other, they react, they bind. And so this has led to this whole idea of facilitated diffusion, where maybe proteins slide on DNA, and that's how they and that can accelerate the target search. So that was one of the things we were looking at for clustering of CTCF. There, we know that CTCF and cohesin bind the same sites by ChIP-seq. But technically, from a ChIP-seq experiment, you cannot know whether they bind at the same time or whether they bind exclusively. So it's possible. I mean, I think we now know that that's not the case, but it is in principle possible that CTCF binds alone and cohesin binds alone, but they bind to the same sites, or they could bind together. And so what we wanted to do was to use super resolution palm imaging to map all the single molecules of CTCF and cohesin in the nucleus, and then ask at the single molecule level, do they co-bind? And that was one of the things we found in the 2017 paper. They do co-bind. Then the other thing we found was that both proteins form small clusters, and a few molecules, dozens of molecules of CTCF form clusters. And this was interesting. And of course, it's hard to say if this is the mechanism, but one of the questions in the field, I think, has been, okay, cohesin can extrude loops. It appears to extrude very quickly, maybe a kilobase per second or so, at least in vitro. And cohesin is on the order of 50 nanometers, and CTCF is probably on the order of 3 to 5 nanometers. So how does something very small reliably stop something extremely fast that moves very quickly, right? Like, it's, if a person walks in front of a train that's moving very fast, that person is not going to stop the train. And that's sort of an analogy for CTCF blocking cohesin. And so clustering there may have a role. And this was, sorry, yeah, go
ahead. Yeah, because I was just wondering, right, how to analyze the CTCF sites by microscopy. How many sites are there? I mean, how much, how much space is between those dots that you can analyze them?
Yeah, so that's one of the beauties of super resolution microscopy, is that you have, in principle, unlimited precision. Practically, I think our localization precision is about 20 nanometers in these experiments. So that's the equivalent of two nucleosomes. So we can localize each molecule plus minus 2 nucleosomes. And the key principle of super resolution microscopy is that, if you take an image of all the CTCF molecules in a cell, just going to light up the whole nucleus. But with super resolution microscopy, we can we can separate in time what we cannot separate in space. And then we can resolve temporally every single molecule and then resolve it. And the clustering led us on a whole detour, because then when we found the clustering, we did a bunch of control experiments to make sure the clustering was real. So, for example, one of the things we did is we labeled the CTCF protein 50-50 in 2 different colors, and then we did 2 color form. And then, if the clustering is real, you would expect the clusters to be half one color, half the other color. And if the clusters were just due to blinking, you would expect it to be either fully one color or fully the other color. So that all checked out. But one of the things we wanted to do was to have a biochemical orthogonal validation of it. And so we generated a cell line where, in mouse embryonic stem cells, where one of the 2 alleles of CTCF is flag tagged and the other allele is V5 tagged. And so now, we have 50-50 flag and V5 tag endogenous CTCF. And then we could do co-immunoprecipitation experiments. So we pull on the flag tag, the V5 tag comes down. We pull on the V5 tag, the flag tag comes down. So that at least confirms that CTCF self-interacts. And then that led us to a whole detour, because then one of the things we did was that we thought, okay, let's see if it's DNA dependent, and DNA's treatment doesn't have such a big effect. But when we treat with RNAs, the co-IP is almost completely lost. And so that was a little surprising. But there was actually a paper from Danny Reinberg's lab in 2014 where they had found the same thing. We only found that after we did the experiment. We should have read that first. But then we thought, okay, so that's interesting. That's not just something we are seeing. And in that paper, Danny Reinberg's lab had shown that a C-terminal region of CTCF, so not the DNA binding region, but a C-terminal region, was necessary and sufficient for RNA binding. So then we deleted this region endogenously in CTCF, and the cells were relatively, like, the growth rate went down, so they're relatively sick. We did micro-C and ChIP-seq, and we found looping phenotypes. So a substantial number of the CTCF loops were lost. In some cases, it was lost due to loss of CTCF and cohesin binding. But we also, so that's easy to explain, right? If you lose CTCF and cohesin binding, you lose the loop. But there was a second class of loops where there wasn't any detectable change in CTCF cohesin binding by ChIP, but the loop was still lost. And so we still don't fully understand that, but one possibility is that it could involve clustering, because we also saw that when you delete this RBR of CTCF, clustering is reduced. But I think it's still an open question, and it's hard to study, right? Because we don't know how to abolish CTCF clustering but keep all other aspects of CTCF the same in the cell.
That's true, and it's also very interesting that many chromatin binding proteins rely on RNA, right?
That's right.
It's also an interesting factor. So you already mentioned it, and you wanted to move on to when you set up your own lab, and there you continued to focus on CTCF, cohesin, and genome organization by extending the previous imaging studies, right? I mean, I like imaging myself because I've done it in my PhD, and I'm always fascinated by fluorescence. But that's just a detour. And maybe you can talk about the transition from the lab with two mentors, I guess it was, then to your own lab. How did that happen?
Yeah. I mean, I suppose so. I had a great time in the Teach and Darsag lab, and I was also fortunate to have some really great colleagues there. I worked very closely with Stanley Shi, who together with Ollie Rando developed micro-C, and Claudia Catoglio, Mustafa Mir, Sheila Teves, Sasha Chong. There was some really great colleagues there. And then applied for lots of jobs, did a bunch of interviews, visited places, and then eventually chose to go to MIT. I think the transition was relatively natural, because I think the core interest of my postdoc lab is really in transcription and maybe less so distal gene regulation. And so that was the thing that I wanted to pursue in my.
So it was no issue that you took the work with you, right?
No. That was all very, very fine, and Teach and Xavier were very supportive of that.
Okay.
And so then so one of the things that I did as a postdoc was look at the dynamics of the CTCF cohesin protein, and we found that they bind chromatin with residence times on the order of minutes to tens of minutes in the case of cohesin, and so that suggests that the loops might also be dynamic. But it is an imperfect argument. It is an indirect argument, right? Because it could be that when a CTCF unbinds, it gets replaced by another one. Or maybe like a cluster of CTCF is bound. So you can have loss of CTCF individual molecules, and it doesn't matter because there's still enough there. And so we really wanted to directly quantify what is the lifetime of one of these chromatin loops. And so we used, classic approaches where you put arrays of binding sites near the two anchors of the CTCF sites. So we used a TED operator array on one side and these PAR-BPAR-S systems that Kirsten Bristecki's lab has developed. And then we, express a fluorescently labeled DNA binding protein. And then we, you can use 3D distance as a readout of looping, because the TAD, the CTCF loop we looked at is a 505 kilobase loop in mouse embryonic stem cells that was originally described as being CTCF dependent by Elso de Wit. And it's a very nice TAD because it's very simple. There's, one strong dot. There's a couple of genes around, but they're not expressed. So it's sort of the hydrogen atom of TADs, right? It's, if we're going to understand a TAD, start with the simplest one. And so then, we can use super resolution live cell imaging to get the XYZ coordinates of the two anchors. And then when it's looped, the distance should be short. And when it's unlooped, the distance should be large. And so that was really the core idea. And I should say this was in close collaboration with Christoph Seckner's lab and Leonid Meni's lab. And then it was led by Michele Gabriele, Hugo Brandau, Simon Busse-Holz, and Esmeda Jar in the lab. And so we tagged the anchors of the loop. We did a whole bunch of control experiments to make sure that the tagging did not affect anything. So we did, micro-seq to make sure that the loop was unaffected. We did chip-seq to make sure the CTCF cohesin binding was not unaffected, and so forth. So we did a bunch of controls, and now we could then visualize looping in real time.
So you say that the genes that are around are unexpressed. So the movement of this loop was then a function of what?
Yeah. So because we tag the CTCF sites. Yeah. So so then I think the two things that contribute to movement are chromatin diffusion, right? Because chromatin just has a certain diffusivity, and then loop extrusion.
Mm-hmm.
And then then we have these 3D distance trajectories as a function of time. And then the key advantage of live cell imaging is that microscopy measurements are noisy, but noise is uncorrelated in time. So from a single measurement, like for example in fish, right? Sometimes you're going to overestimate the distance, underestimate the distance, overestimate the distance, underestimate the distance. And so if you just have one measurement and it's far, it could have been contact, but you overestimated it. Or if it's low, it could have been not contact and you underestimated it. But the advantage of live imaging is that noise is uncorrelated in time. So when you have many measurements as a function of time, you can statistically make out, are you looped over time? And so we developed this Bayesian inference method that we call BUILD for Bayesian inference of looping dynamics that allows us to very rigorously determine when are you looped and when are you not looped. And we found that the particular loop that we looked at, it's looped on the order of 5% of the time, and it has a loop lifetime on the order of 20 minutes. And so that means that if you look at a TAD in high C, you see a nice triangle, you see a nice dot, and it looks very orderly. But this is this is an average over typically millions of cells. And so at the single cell level, these loops form and break throughout the cell cycle, and only looped on the order of 5% of the time. And then you might ask, well, okay, if the loop is so rare and so dynamic, is that really consistent with what we see in high C? And it actually turns out it is. If you simulate very stable and static loops using polymer simulations, you cannot reproduce the high C data. But if you simulate dynamic loops, you can reproduce the high C data. So one of the things we were able to do in that paper was we were able to fully parameterize the TAD. So put numbers on all the parameters, like how many cohesins extrude, what's the processivity, what's the probability that a CTCF boundary will actually block an extruder, what's the lifetime, what's the looping probability. And we found that although the fully looped structure is relatively rare and infrequent and only form on average once or twice per cell cycle, that doesn't mean that extrusion is rare. We actually found that 92% of the time, this particular loop is partially extruded. It's just that cohesin will often extrude for a while and then fall off before it can reach the CTCF boundaries. And I should also just say here that Luca Giorgetti's lab found something similar in a synthetic CTCF loop, and there's a preprint by Thomas Sabaté and Edouard Bertrand, Christophe Zimmer that also finds something similar in a human cell type that came out afterwards.
Mm. So I guess all this dynamics is also influenced by cohesin being this motor protein that constantly moves DNA, probably, right?
That's right. Yeah. So I think one of the things I think is important to take away from this is that if we think about the function of TADS and extrusion, I think it might be more about the journey than about the destination. So if you imagine a very static system where you have these very static loops that are always present, then the only thing that allows pieces of DNA to find each other is diffusion. And diffusion is very efficient at short length scales, and it's extremely inefficient at long length scales. So we had a preprint earlier this year when we applied MinFlux and a few other techniques to really very carefully quantify chromatin dynamics. And one of the things we find there is that if you look at the diffusion, if you take let's say an enhancer and a promoter, and that's currently 50 nanometers apart, and move them 500 nanometers apart, so increase the separation by a factor of 10. The first passage time will increase by about 5 million. Mm. So it's much, much slower. And so I think one of the things that extrusion does is that it reduces the dimensionality of the search. Instead of doing random 3D diffusion, you do active 1D tracking along the chromosome. And so this allows enhancers to search for promoters on the same chromosome. And if it was static, then it would only happen once. But by having it be dynamic, you do it falls off, you extrude, you fall off, you extrude, you fall off. That allows enhancers to constantly sample promoters that might be further apart. And so this is likely necessary. So having this dynamic process is likely necessary for distal enhancers to find their target genes. Mm-hmm. And indeed, I think there's emergent papers from several labs in the field showing that cohesin is probably not very important for enhancers that are close to the promoter because diffusion is very efficient at those scales. But once you go far away, whether that's 100 kb or more, then cohesin extrusion is really important for enhancers finding their target genes.
So another thing that you worked on is how double-strand break repair is connected to DNA loop extrusion. So I guess the important thing for DS breaks is that the machinery can access the double-strand breaks. And if cohesin comes in and maybe blocks it, it can be difficult, right?
Yeah, absolutely. And I think cohesin, several labs have suggested that for homologous recombination in replicated chromosomes, cohesin can help with the target search as well. In this paper, we specifically wanted to focus on G1 double-strand break repair. So before replication, where typically things are repaired by non-homologous end joining. And there, non-homologous end joining you can break down into two key steps. So after the double-strand break has happened, right, the two DSB ends have to get back together, and then they have to be repaired and ligated. And the first step of getting back together is called synapsis. Mm-hmm. And there had been a paper from Tom Mistelli from at this point almost 20 years ago where they induced double-strand break repair, double-strand breaks, and then they saw that sometimes the ends can move quite far apart, hundreds of nanometers apart. And once the ends have moved very far apart, a question is, well, how do they actually come back together, right? Because diffusion is not intelligent. Diffusion doesn't know which way to go, right? So diffusion is just as likely to move them further apart as back together. And it's estimated that each cell in your body has about one double-strand break event every day. And so if you just look at all the cells in your body, right, it's not enough that the two ends come back together 99% of the time because you have billions of cells in your body. The fidelity has to be very high. And so in that paper, and I should say this was led by Harvey Gang and Hugo Brandau, in that paper we wanted to explore how could loop extrusion help? And I think it can help in two ways. So one is that if a lot of the genome is extruded into loops, then very often a break will happen inside a cohesin loop. And now, because a cohesin is bridging the two pieces of DNA, they are prevented from diffusing too far apart. And so this prevents this sort of diffusive catastrophe. And then the other thing is that for bringing the two ends into close proximity, we show that extrusion can also facilitate that. So this was a fully theoretical paper. So this was and this was actually a project we started because of COVID. So I started my lab February 1st, 2020, and 6 weeks later everything shut down. And so Javi was actually working on another project, and then this was sort of a COVID project. But one of the things Javi did was he using just analytically, using analytical math, right? He derived expressions for how extrusion should affect all of these, and then he used polymer simulations to see what are the parameters that will facilitate this. And so one of the things he found was that extrusion is likely very important for both preventing this sort of diffusive catastrophe where the DSBs move too far apart, and also for bringing them back together.
Yeah, that was actually what I was thinking when I read this, right? Bring those broken pieces together is similar to bringing the CTCF sites together, right? So, but it's not like that. That's you have proteins on the breaks that will then bring it back together.
Yeah, so I mean, there are of course lots of repair proteins that get recruited to the ends, and that really help them stick together. But there is a delay, right? Because if in the nanosecond after the break has happened, they're not going to be there. So it takes a while to sense that there's a break and recruit all the proteins. And during this time, there is a probability that the ends move apart. And so we think extrusion helps reduce the likelihood of that happening, and also help bring the ends back together.
So historically, chromatin architecture has been investigated, and you already alluded to this, by using high-C, 3C, more recently micro-C, which is a very elegant method. But you developed a new assay based on micro-C to generate even deeper genome, 3D genome maps. Can you talk about this approach?
Yeah, so we developed this method called that we call region capture micro-C, and the motivation was really that despite sequencing getting much cheaper, we are just way off 1x coverage of the 3D genome. And to illustrate this, if you do 1D genomics like a tag-seq or chip-seq and so forth, right? If you want 1x coverage of the human genome at nucleosome resolution, you need about 22 million reads. And so 1x coverage is very easy, right? Like if someone says, I have achieved 1x coverage at tag-seq, few people would be impressed, right? It's very easy, and at this point, it's very cheap. I think at our sequencing facility, this will cost maybe on the order of $20. But in 3D genomics, the data format is not a vector like it is in 1D genomics, it's a matrix. And so 1x coverage at nucleosome resolution for 3D genomics, you need to take 22 million and square it. So now you get 4 times 10 to the power of 14.
Because because you have like two pieces together in one read, right?
That's right. Yeah, because for every single bin, you're asking how often does it interact with the entire rest of the genome. Yeah. And so that's why for 1x coverage, you need the genome square. And so, one of the landmark high-C papers was the Rao-Huntley paper from 2014, where they achieved 5 billion reads, which was amazing, and it's a landmark paper in the field. But 1x coverage at nucleosome resolution would be 4 times 10 to the power of 14. So you're still let's see, 10 to the power of 5 off. And then the core idea is, well, if the sequencing cost scales with the genome squared, if instead of capturing 3 gigabases, you capture 3 megabases, then you reduce the linear space by a factor of 1,000. So a factor of 1,000 squared is a factor of 1,000,000. So now the sequencing cost actually goes down by a factor of 1,000,000. So whereas 1x coverage of the human genome at nucleosome resolution would cost billions of dollars, now if we just capture 3 megabases instead, it costs a couple of thousands of dollars. And so we can just achieve very, very deep maps by not capturing the whole genome. And this was work led by Virat and Miles in the lab. And, you know, I was fortunate to work with Stanley Shi when I was at Berkeley. Stanley Shi and Oliver Brando developed microC. So we learned microC from Stanley. And then, you know, we set it up in the lab. The genome-wide microC protocol is reasonably efficient, but it was not quite efficient enough for very deep capture. So we spent about a year and a half troubleshooting it and optimizing it. I think we changed 11 steps in the protocol, and so then we could get very, very high library complexity that's suitable for this very deep capture. And so now, you know, we can get 100 times, 1,000 times deeper maps than you know the best genome-wide maps without spending much money on sequencing. And that's really revealed both new 3D genome structures, but also just if we want to do quantitative work, like say, understand how much loop strength scales with gene expression, you really need very deep maps in order to accurately quantify the loops. So we're very, very excited about reaching capture microC, and it's been nice to see quite a few other labs also taking it up and having good results with it.
So maybe I missed this or didn't understand it, but how do we achieve this reduction?
In sequencing costs?
Yeah, no, because are you not like looking genome-wide, or how would?
So, sorry. Yes. That's a key part that I skipped. Yeah. So we use that's a key part that I skipped. So we use a capture. So and we are not the first people to do that. People have been using region capture and promoter capture and enhancer capture, Hi-C in the past as well. So you can buy biotinylated DNA probes. We use 80 base pair probes from various companies like Twist and Roche. And then you can tile a region, and then you buy the probes. They give you the probes, they give you a capture protocol, and then we just do that. Oh. We've been using Twist. Yeah, okay. Yeah, sorry. I
so it's basically limiting the starting points.
Yeah. And then so it's sort of the trade-off is quantity or quality, right? You can either do low resolution genome-wide, or you can do very high resolution region capture. Okay, yeah. And in some cases, it makes sense to do one, and in other cases, it makes sense to do the other. But I think one thing that I think is very important is that in the 3D genome field, we are just massively undersampling. We are way off 1x coverage, and so I think there are still quite a few structures to be discovered. And if you go through the history of the Hi-C field, new discoveries have actually often followed improvements in protocol and especially sequencing. So, you know, the original Hi-C paper, I think from 2009, I think discovered the AB compartments, I think had on the order of 8 million contacts. And then in 2012, Bing Ren's lab and Edith Hurd, Fishnora, Jesse Dixon, and a few other people. So they achieved, I think, on the order of 450 million contacts, and now they could see the tests. And then in 2014, Suhas Rao, Miriam Huntley, Eris Lieberman Aiden achieved 5 billion contacts, and now you can see the loops for the first time. So every time you sort of improve the depth by 10 to 100 fold, you tend to discover a new pattern in the maps. And we actually did the same. So with region capture microC, we reached the equivalent of 317 billion genome-wide contacts, and we observed these microcompartments. And one of the advantages of microC compared to Hi-C is that it's much more sensitive to enhancers and promoters. Hi-C is very good at detecting CTCF loops, but less so as dysregulatory elements. And so unexpectedly, we found these highly nested focal interactions between enhancers and promoters when we applied region capture microC. So in the contact map, it looks like a grid of dots, suggesting fairly promiscuous interactions between enhancers and promoters. And this was also one of the things, one of the reasons why we wanted to develop this method is that we've been very interested in understanding the principles of enhancer promoter selectivity. So to what extent do enhancers selectively loop to some genes versus other genes, which could be a key aspect of selectivity in gene regulation. And so what we found in that paper is that the interactions are fairly promiscuous, so that the selectivity, it's not binary. Our data is very consistent with selectivity, but it's maybe a factor of 2 or 3. This enhancer likes this promoter by a factor of 2 or 3 more than this other promoter. So it's exciting to see this microcompartment pattern.
Yeah, very interesting. I guess you also use this method then to look at the function of ZNF143 and ZFP143, and looking more into the function of this protein. So why exactly this protein? That's a funny story. So we've been very interested in understanding the selectivity of looping, right? So one of the things we have found and other people have found is that CTCF and cohesin are extremely important regulators of genome structure. And if you get rid of CTCF and cohesin, all the CTCF and cohesin loops go away. I mean, that's also why they're called CTCF and cohesin loops. But enhancer promoter interactions are for the most part unaffected. And that was also one of the things we show in the RCMC microcompartment paper is that when we deplete cohesin, there's like on the order of 5-10% of enhancer promoter interactions changed by a factor of 2. But for the most part, enhancer promoter interactions are retained. And so that means that CTCF is important for some, especially the enhancers and the promoters that are bound by CTCF. But there's a whole class that are not regulated in that way, and therefore the mechanism has to be something else. And so that then raises the question of what are these other looping factors? And we actually don't know a lot about the other looping factors. So in Drosophila, for example, there's at least a half dozen insulator proteins, and CTCF is probably not one of the is not number one. But in mammals, we don't know as much. And so there was prior work suggesting that ZNF143 was a looping factor, and so there was some papers that have found that it was a looping factor independent of CTCF, and there was some that had found it was a looping factor dependent on CTCF. And so we thought, but so there had been several papers that motivated us to study it, but there hadn't been this sort of very clean experiment where you put a dacron on the protein, you deplete it, and then you do deep micro-C. Or high-C, which I think is really that's sort of the gold standard for testing. How strong of a looping factor is it? And so we did that. Well, or Dominic Narducci, the grad student in the lab who led this project, did that, and we depleted ZNF143 and mouse embryonic stem cells, and not too much happened. The effects were very modest. So then we thought, okay, well. Maybe a dacron is not complete enough. So then we switched to, we added a D tag instead of an AID tag. We repeated the experiment. Nothing much happened. And we thought, well, okay, maybe it's a weird clone. So then we made more clones, and we did extremely deep micro-C, right? These are some of the deepest micro-C experiments that have been published. So our detection sensitivity is very good. And, we didn't really see much of a change, and we analyzed the data in every way we could to try to find that it was a looping factor. But the effects were modest. Even on anchors that are bound by ZNF143, the effects were very modest. Then we thought, okay, well, most of the prior studies have been in human cells, so let's repeat the whole thing in human cells. So then we did dacron experiments in human cells. Deep micro-C, not much happened. So no matter how we sliced the data, it just did not look like a looping factor. And then at a conference, I had lunch with Elsa de Wit, and one of the first questions he asked me was, what do you think about ZNF143?
I don't know how he knew we were working on it, but it turned out he was working on the same thing, and he had done the same experiments, and he had found that it was not no matter how he sliced the data as well, it was just not a looping factor. And so, and it was really nice to be able to coordinate with ELSO, because if we really tried to find that there was a looping factor, and no matter what we did, it just was not. And so that makes you, a little nervous. But he found exactly the same thing. And what appears to have happened is that ENCODE, they did ChIP-seq of hundreds of proteins. And, obviously the service, ENCODE has just been an amazing contribution to the genomics field. It's just transformed what we can do. But whenever you work with antibodies, sometimes you have an antibody that misbehaves. And it turns out the antibody they use for ZNF143 cross-reacts with CTCF. And therefore, the reason why the people thought ZNF143 was a looping factor is because CTCF is a looping factor, and the ZNF143 antibody cross-reacts with CTCF. And so if you look at all the ChIP-seq data that's around, if you look at if you use the antibody that ENCODE used, ZNF is enriched at loops. But if you use other ZNF143 antibodies, it's not. So this whole thing appears to have been an antibody artifact, and this was really this was also key work. This was key work from ELSO that we did. Okay. So then we coordinated, we co-submitted these papers. It was nice to have strength in numbers. So ZNF143 is a very interesting protein. Actually, it 7-zinc finger DNA binding protein. It's a transcription factor. It binds to promoters. It doesn't really bind to enhancers. It regulates a few hundred genes. We depleted it and did pro-seq. So it's a very interesting and important transcription factor. From a dynamics point of view, it actually has the longest residence time of any transcription factor ever measured to our knowledge. So it binds so stately to DNA that we can't even totally accurately measure it. It appears to have a residence time of more than 20 minutes. So it's a very interesting protein, but it's unfortunately not a looping factor, which we had kind of hoped, right? Because we need ways of explaining how do all these CTCF independent loops form, right? It would be very attractive if there were other looping factor proteins. But ZNF143 appears to have been a red herring and an antibody artifact. Yeah.
Interesting is that you can at some point find those antibody artifacts, which is interesting. So there is also about 4 bioRxiv papers out there that are waiting for peer review, I guess. So do you want to talk about maybe any of those?
Yeah, I mean, I can briefly talk about some of them. Yeah, so one preprint we have is on absolute quantification. So we like numbers a lot, and Hi-C and Micro-C, obviously incredibly powerful methods that have taught us probably most of what we know about genome architecture. But it is all relative numbers, right? It's relative sequencing reads. And so what we thought was, okay, we know how to do absolute quantification, like this loop is looped X% of the time by super resolution live cell imaging, and we know how to do Micro-C. So now, if we do Micro-C on cell lines where we have also quantified some loops, we should be able to calibrate the Micro-C and do absolute quantification. And so that's really what we did in that paper. And so then we were able to estimate the absolute looping probability of all loops across the genome. And so we quantified 36,000 loops in that paper. And we find that in general, loops are quite rare. So we found the average looping probability to be just over 2% in mouse embryonic stem cells. And CTCF loops are stronger than most enhancer promoter loops. I think the average CTCF loop is on the order of 3%, and the average EP loop is on the order of 1%. And the strongest loops in MESCs are on the order of 20-25%. Actually, the famous SOX2 enhancer promoter loop is one of the stronger ones. Okay. There is, of course, a little bit of a sampling bias here, because if you only analyze the strongest 1,000 loops, the average is higher. And if you analyze 36,000 loops, the average is lower. But it generalizes that loops are rare, and we provide all that data as well. We have a preprint on mitosis that actually just was published last week on Friday. So it just came out in Nature Structural Molecular Biology, where the motivation was okay. So we found these microcompartments using region capture micro-C, these sort of nested focal interactions, and then we tried to perturb them. We got rid of loop extrusion, and they were for the most part unaffected. And we inhibited transcription because, they mostly form at active enhancers and promoters. So maybe they are downstream of transcription. So we inhibited Pol2. And again, 5-10% change by a factor of 2, but it's relatively modest. So then we thought, okay, let's go to another system where we can try to understand how they form. And the pioneering work from several labs, including Joop Dekker, Gert Blobel, and several other people have looked at 3D genome structure throughout the cell cycle. And one of the things they found is that by high C, if you look at mitosis or chromatophase, almost all high C observable genome structure is lost. So AB compartments are lost, ads are lost, CTCF loops are lost, transcription is for the most part shut off, and so forth. And so that led, this has led to this idea that, you erase genome structure every cell cycle, and then you have to rebuild it de novo as cells into G1. So we thought, okay, if we want to understand how these microcompartments form, then we should start at mitosis, because then we can watch them form from chromatophase as cells go into G1. And so we collaborated with Gert Blobel's lab, and Ed Bannigan and Leonid Mirny to look at this, and we found the opposite of what we expected. So we expected to see no pattern genome structure in mitosis, and then structure forms as you enter G1. But what we found was that microcompartments are present in mitosis. And so the RCMC data in mitosis is completely consistent with high C at the level of AB compartments, TADs, CTCF loops. You also see complete loss of those in mitosis. But because we have about 100 to 1,000 times higher resolution with region capture micro-C, we could see these microcompartments in mitosis. And so we see these nested focal interactions between enhancers and promoters in mitosis. And they actually peak in strength at around an telophase. And then a lot of them get washed out as you enter G1 phase. And this was surprising and not what we expected. But, you have to do the experiments in the forward direction, but you can only understand why in the backwards direction. In retrospect, I think it makes sense for a couple of reasons. So one thing is that previously, Gert Blobel's lab had discovered this transcriptional spiking phenotype, where on the order of half of all genes have this transient spike as they exit mitosis. And so if you have fairly promiscuous enhancer-promoter interactions in mitotic chromatin, that might explain it. The other thing is that microcompartments, we think that enhancers and promoter interactions are fundamentally affinity mediated. So we think that it's stickiness. And we also think that explains why they're robust to loss of loop extrusion. So you can imagine that most enhancers and promoters are bound by transcription factors, right, and various co-activators and so forth. And most of these proteins have intrinsically disordered regions, low complexity domains that engage in multivalent interactions. So you would expect all enhancers and promoters to be a little bit sticky with each other. And if you simulate that, you can fully reproduce this sort of grid of dot-like pattern that we see. And then you might ask, okay, so biophysically, how are affinity-mediated interactions regulated? In polymer parlance, this is called block copolymer microphase separation, which is a mechanism that has been known for almost 100 years. It's not the same as liquid-liquid phase separation. It's just saying that if segments of a polymer are a little bit sticky with each other, they're more likely to interact. And so getting a little bit into the biophysics of it. So, whether something happens depends on the Gibbs free energy, which is related to the enthalpy minus the temperature times the entropy change. And we mostly in the 3D genome field think in terms of enthalpy, right? Like how strong are the affinities? But entropy is also very important. And whenever you form an interaction or a loop, right, you go from a more disordered polymer to a more ordered polymer. And so there's an entropy cost to forming specific interactions. And the configurational entropy of a decompacted polymer is very high, and the configurational entropy of a compacted polymer is very low. And mitotic chromosomes are very compacted. And that means that the entropic penalty you pay for forming interactions is just much lower. And so one of the things we show in that paper is that if you change the compaction by a factor of 2, make chromosomes 2 times more compact, you can increase the strength of affinity-mediated interactions by a factor of 6. So it's quite strong. And so we think that mitotic chromosomes are actually kind of perfect for forming affinity-mediated interactions, microcompartments, EP interactions, because around an telophase, loop extrusion is relatively modest and chromosomes are very compact. So this is the perfect environment for forming these promiscuous interactions. And then probably what happens as cells enter G1 is that CTCF and cohesin and other factors come back on chromatin. They begin making tads and loops, and then they prune some of these promiscuous microcompartmental interactions, and they begin to establish order. And I should say this paper was co-submitted with a paper from Joop Dekker's lab, where they find similar things in a different system, and that should be coming out soon as well.
But those loops are then mainly a function of linear distance, right? Because the chromosomes are folding because of their linear sequence.
Yeah, that's right. Yeah, but because mitotic chromosomes are so compact, it's actually easier to form long-range interactions in mitotic chromosomes. So one of the things we find is that the promiscuous enhancer promoter dots in mitotic chromosomes, it's the more long-range ones that are preferentially washed out as cells into G1.
Okay.
And I think it's sort of that's probably also one of the functions of loop extrusion is that in the absence of loop extrusion, chromosomes would just kind of go into their thermodynamic equilibrium, which would be all the sticky parts going together, and loop extrusion enforces mixing to prevent.
Okay. So imagine you're due to submitting a grant proposal tomorrow. What would you have written into this? Would this be more of the micro-C experiments?
Well, yeah. Well, I mean, actually, this is a very busy grant month, so I think I'm submitting 5 or 6 grants this month. So
there you go.
quite a few different things. But I think broadly, we're interested in 3 key questions. So one is enhancer promoter selectivity. A typical megabase in the human genome has on the order of 10 genes and 100 enhancers. We know enhancers can activate across 1-2 megabases. So combinatorially, there's just a huge number of possible interactions. But we know that gene regulation is specific. So what are the mechanisms that ensure enhancer promoter selectivity? It's probably going to be, rather than one mechanism explaining everything, it's probably going to be many different mechanisms, each explaining factors of 2, 3, 4, 5. But what are those mechanisms? 2 is the spatiotemporal mechanisms of enhancer promoter interactions. So, enhancers were discovered in 1981, and the classic idea is that you have a stable loop between the enhancer and the promoter. But we still don't know, right? So in space, how close do enhancers have to get to their target genes to activate? The classic model is contact, but there's been various other proposals over the last 5-10 years suggesting that maybe you could have action at a distance. This could be mediated by condensates or gradients. So we still don't know how close enhancers and promoters have to get in space. And in time, we also don't know, right? I mean, we know that the CTCF loops last on the order of 20 minutes, at least in the cell types we've looked at. But we don't know what it is for enhancer promoter interactions. We know that enhancer promoter loops are weaker than CTCF loops. So they either have to be, if they are long-lived, they have to be very infrequent, or if they're frequent, they have to be very short-lived. But we don't really know. So in space and time, what do functional enhancer promoter interactions look like? And then a third question is, how does the genome misfolding cause disease? that we know there are many examples where we know the genetics, where genetically we know that loss of a CTCF site can cause aberrant enhancer promoter interactions. Or we know that, for example, the STAG2 subunit of cohesin is among the 12 most mutated genes in cancer. So there are many connections between disease and genome structure, but yeah. But in each case, how does it work and what can we do about it?
Okay. So for the last almost 1 hour, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
Very briefly, there are 2 preprints that we put out in May that we're excited about. So one is using machine learning to map genome structure at very high resolution. As we talked about, right, we can either do low resolution genome-wide micro-C, or we can do ultra high resolution local RCMC. But what we want is ultra high resolution genome-wide maps, right? And so we've developed this. This is work from Clarice Hong and Fan Feng in collaboration with Qilu's lab. We've developed an ML model we call Cleopatra that trains on genome-wide micro-C, fine-tunes on RCMC, and then predicts RCMC resolution, genome-wide 3D structure. That seems to work quite well. So that's one thing we're excited about. We've also been in work from Matteo Masaka, Simon Grosse-Holz, Dominic Narducci, Jessica Matias. We've been applying a new microscopy technique called MinFlux to look at chromatin dynamics. And we just asked a very basic question. How does chromatin move? And how long does it take 2 pieces of DNA to find each other? And this has been very difficult to answer because to look at this, you need to look at extremely fast timescales and you need to look at extremely long timescales. And so by integrating MinFlux, single molecule tracking, and super resolution live cell imaging, we're able to track chromatin dynamics over 7 orders of magnitude, a range of 36 million actually. And this reveals chromatin to be very, very subdiffusive. And this would probably be a longer discussion, but one of the key findings is that 2 pieces of DNA that are close, let's say within 100 or a few hundred nanometers, they are just going to bump into each other extremely efficiently. So contact is kind of inevitable. And the flip side is that things that are far apart, more than a micrometer or so, it's going to be almost impossibly slow for them to find each other, maybe more than a cell cycle. So contact at short scales is inevitable, and contact at long scales is almost impossibly slow, which we think have many implications for basically any process that involves 2 pieces of DNA finding each other, like DNA repair, enhanced promoter interactions, recombination, and so forth.
Yeah, this will be a nice discussion once the paper is then finally published.
Yes.
So thank you, Anders, for your time and for being on the show.
Yeah, thanks so much for having me. It was great.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.